All life needs a constant input of energy. Animals get their energy from eating other organisms. Plants make energy and synthesize sugars through photosynthesis. Photosynthesis is basically respiration found in heterotrophs run backwards powered by light. Plants need to collect light energy and transform it into chemical energy and store light energy in a stable form to be moved around the plant or stored. Plants need to get building block atoms from the environment. Plants need to produce all the organic molecules needed for growth. Plants obtain sunlight from leaves, CO2 from stomates, H2O from roots, and nutrients from roots. Stomates allow for transpiration and gas exchange. A typical mesophyll cell has 30 to 40 chloroplasts, each about 2 to 4 microns by 4 to 7 microns long. Each chloroplast has two membranes around a central aqueous space called the stroma. In the stroma are membranous sacs called thylakoids. They contain chlorophyll molecules, electron transport chain, and ATP synthase. They may be stacked in columns called grana. Photosynthesis contains light reactions that convert solar energy to chemical energy and the Calvin cycle, which are light-independent sugar-building reactions. Light reactions contain the electron transport chain, which have proteins in organelle membrane, electron acceptors such as NADPH, proton gradient across inner membrane, and the ATP synthase enzyme. In the electron transport chain of cellular respiration, mitochondria transfer chemical energy from food molecules into chemical energy of ATP, with NADH as the input and ATP as the output. In the electron transport chain of photosynthesis, chloroplasts transform light energy into chemical energy of ATP. Remember, photosynthesis is anaerotic. The electron transport chain is driven by light energy. In both the sunlight of photosynthesis and the breakdown of glucose in respiration, they both move the electrons, run the pump the protons, build the gradient, drive the flow of protons through ATP synthase, bond P to ADP, and generate ATP. Chlorophylls and other pigments are embedded in the thylakoid membrane, arranged in a photosystem, and have a structure-function relationship. Photosynthesis gets energy by absorbing wavelengths of light. Chlorophyll A absorbs best in red and blue wavelengths and least in green. Accessory pigments such as chlorophyll B, carotenoids, and xanthophylls with different structures absorb light of different wavelengths. Photosynthesis has two photosystems. Photosystem 2 has chlorophyll A and absorbs 680 nanometers of wavelength red light. Photosystem 1 has chlorophyll B and absorbs 700 nanometers of wavelength red light. Photosystem 2 absorbs light. Excited electrons pass from chlorophyll to the primary electron acceptor. They need to replace electron in chlorophyll. An enzyme extracts electrons from H2O to supply them to chlorophyll. This reaction splits H2O into two H plus and O negative, which combines with another O negative to form O2. O2 is released in the atmosphere. Chlorophyll absorbs light energy, also known as a photon, and this moves an electron to a higher energy state. Electron is handed down chain from electron acceptor to electron acceptor. In this process, H plus ions are collected from H2O and also pumped in plastoquinone within thylakoid sac, which flow back through ATP synthase to generate ATP. There needs to be a second photon, a shot of light energy to excite the electron back up to high energy state. The second electron transport chain drives reduction of NADP to NADPH. Light comes in at 2 points to produce ATP and NADPH. O2 came from the radioactive tracer O18, which proves O2 came from H2O, not CO2, because plants split H2O. In non-cyclic photophosphorylation, light reactions elevate electrons in two steps, photosystem 2 and photosystem 1. Photosystem 2 generates energy as ATP, while photosystem 1 generates reducing power and NADPH. In cyclic photophosphorylation, if photosystem 1 cannot pass electrons to NADP, it cycles back to photosystem 2 and makes more ATP but no NADPH. It coordinates light reactions to the Calvin cycle, which uses more ATP than NADPH. In the Calvin cycle, there are 3 steps. 1. Carbon fixation, 2. Reduction, and 3. Regeneration of RUBP. In carbon fixation, a 5-carbon sugar molecule called ribulose bisphosphate or RUBP is the acceptor that binds CO2 dissolved in the stroma. This process called CO2 fixation is catalyzed by the enzyme RUBP carboxylase, forming an unstable 6-carbon molecule. This molecule quickly breaks down to give 2 molecules of the 3-carbon 3-phosphoglycerate 3-PG called phosphoglycerate acid PGA. In reduction, the two 3-PG molecules are converted into glyceraldehyde 3-phosphate. G3P, aka phosphoglyceraldehyde PGAL molecules, a 3-carbon sugar phosphate by adding a high-energy phosphate group from ATP, then breaking the phosphate bond and adding hydrogen from NADH and H+. In regeneration of RUBP, 3 turns of the cycle using 3 molecules of CO2 produces 6 molecules of G3P. However, only one of the 6 molecules exits the cycle as an output, while the remaining 5 enter as a complex process that regenerates more RUBP to continue the cycle. 2 molecules of G3P produced by a total of 6 turns of the cycle combine to form 1 molecule of glucose. Glyceraldehyde 3P is the end product of the Calvin cycle and an energy-rich 3-carbon sugar. It is an important intermediate, producing carbohydrates from glucose, phospholipids, fats, waxes from lipids, proteins from amino acids, and DNA and RNA from nucleic acids. Ribisco is an enzyme which fixes carbon from air. Ribulose bisphosphate carboxylase, the most important enzyme in the world and definitely the most abundant. The returns of the Calvin cycle produce 1 G3P. 3 CO2 yields 1 G3P. 6 turns of the Calvin cycle produce 1 glucose. 6 CO2 yields 1 glucose. 18 ATP plus 12 NADPH yields 1 glucose. Photorespiration and the oxidation of RUBP causes a short circuit of Calvin cycle. Loss of carbons to CO2 reduces production of photosynthesis. If photorespiration could be reduced, the plant would become 50% more efficient. C4 and CAM plants reduce photorespiration by separating carbon fixation from the Calvin cycle. C4 plants physically separate carbon fixation from the Calvin cycle by different cells to fix carbon versus where Calvin cycle occurs, and it stores carbon in 4C compounds. The different enzyme to capture CO2 to fix carbon is PEP carboxylase, and there's a different leaf structure. CAM plants separate carbon fixation from Calvin cycle by time of day. They fix carbon during the night and store carbon in 4C compounds. They perform the Calvin cycle during the day. C4 and CAM plants should be seen as variations on carbon fixation because plants had to evolve alternative systems given the limitations of their enzymes and their need to conserve water. C4 plants are a better way to capture CO2. The first step before the Calvin cycle, they fix carbon with enzyme PEP carboxylase and store it as a 4C compound. They have an adaptation to hot and dry climates and have to close stomates a lot and have different leaf anatomy. Examples of C4 plants include sugarcane, corn, and other grasses. C4 plants solve the photorespiration problem by fixing carbon in outer ring of mesophyll cells using a different enzyme, PEP carboxylase, which has a higher affinity for CO2 than O2, better than Ribisco. So it fixes CO2 in 4C storage compounds, oxaloacetate, and malate. It then passes the carbon on by regenerating CO2 in the inner bundle sheath cells for Ribisco to use in the Calvin cycle. CAM plants have an adaptation to hot and dry climates and separate carbon fixation from the Calvin cycle by time of day. They close stomates during the day and open stomates during the night. At night, they open stomates and fix carbon in 4C storage compounds. During the day, they release CO2 from 4C acids to the Calvin cycle and increase the concentration of CO2 in cells. Some examples of CAM plants include succulents, some cacti, and pineapple. C3 problem could be the problem with photorespiration because possibly it had evolutionary baggage. Rubisco evolved in high CO2 atmosphere because there wasn't strong selection against active site of Rubisco accepting both CO2 and O2. Today it makes a difference with 21% O2 versus 0.03% CO2. Photorespiration can drain away 50% of carbon fixed by the Calvin cycle on a hot and dry day. There is a strong selection pressure to evolve better ways to fix carbon and minimize photorespiration. Photosynthesis consists of light reactions in the Calvin cycle. In light reactions, ATP is produced and ADPH is produced, H2O is consumed, and O2 is produced as a byproduct. In the Calvin cycle, CO2 is consumed, G3P, a sugar, is produced, ADP is regenerated, and NADP is regenerated. C4 and CAM plants reduce photorespiration by solving CO2 and O2 gas exchange versus H2O loss challenge. C4 plants separate two steps of carbon fixation anatomically in two different cells. CAM plants separate two steps of carbon fixation temporarily at 2 times of the day, night and day. I hope you learned a lot about photosynthesis. Thank you for watching.